Okay, and our third and final speaker is Aaron Asmus from both Hormel Foods and the University of Minnesota. And Aaron is the Director of Lab Services and Refrigerated Foods Product Development for Hormel Foods. In his current role, Aaron leads research efforts in food safety and quality, product and process development, thermal processing, and analytical chemistry. Additionally, Aaron is a PhD candidate in comparative and molecular biosciences at the University of Minnesota under my guidance. And Aaron's research interest is the microbial ecology of fresh pork and how it affects food safety and quality. And Aaron is going to talk to us today about salmonella in fresh pork and a regulatory update. Hold
Right.
on one second. Let me get this up for you.
Well, thank you, Noelle. First off, I just want to say that as a meat scientist, this is one of the highlights of my career, being asked to present at a swine veterinary conference. So thank you. Very much appreciate the opportunity to present today. That's all right. I can talk. No worries. So what I'm going to talk a bit today about is a little bit about what I have to deal with on the processing side. We're going to talk a little bit about this because of what we do at Hormel Foods. We obviously have a lot of work that we do in pork processing, but we also have Jennie-O Turkey Store. So we're very much exposed to what is going on within the poultry industry in salmonella. And so a lot of the learnings that we have from that will apply to a certain extent to what we have going on and what is coming in pork. And so I'm going to spend half the presentation talking about what led USDA and FSIS to the point where salmonella is now being considered an adulterant in those products. But then to also, in the second half of my presentation, I'm going to talk a little bit about some of the research that we've been doing that I think dovetails really nicely with the two previous presentations. So to talk a little bit about where we've been in the poultry industry. It would have been about 2008 was when the turkey and chicken industries were being held to a performance standard. That was 100% prevalence based. It was positive, negative. It was based on a rolling 52-week average of basically USDA coming in, taking a weekly sample. And you had 52 shots at it, and if you exceeded that prevalence rate, you got put in the penalty box, is probably the way to put it. That was made public information, so our retailers could go look at what our salmonella results were and make decisions as to whether or not they wanted to carry our ground turkey versus our competitors based off of the results of that salmonella testing. And so that has been in place since 2008. And as a result of that, as you can see on the left, looking at chicken carcass rinses and ground turkey on the right, there's been a very steady decline since 2008 in terms of the overall prevalence of salmonella in these two products. I'm going to call out a couple of instances where you see a dramatic spike in 2016 in the carcass rinse, as well as towards the end of 2013 in ground turkey. Those spikes were caused by changes in the way that we were monitoring for salmonella. So in the case of the carcass rinses, we went from taking swabs to actually doing a whole carcass bird rinse. Which was a much better way to be able to try to find salmonella throughout the entire carcass. And then in addition to ground turkey, we went from a 25-gram sample to a 375-gram sample. So you take a bigger bite at the apple, chances are you're going to find a higher prevalence of salmonella. So that's what those kind of odd spikes are in the data. But again, the general trend is that we've done a really good job as an industry driving the overall prevalence of salmonella down in these items. However, this is the actual infection data. So there's a number of infections per 100,000 people within the population. This is CDC data going all the way back to 1996. And as you can see, with exception to a very sharp decline in Campylobacter right off the bat, which is the purple line that you see, we have remained generally flat since 1999 in terms of actually reducing the number of illnesses per 100,000 for not only Campylobacter, but for Listeria, S-Tech, and then the green bar there is Salmonella. So as you can see, we really haven't changed. While we've driven prevalence down, we have not made much of a dent in terms of actually reducing illnesses. In USDA, FSIS, and the Office of Disease and Health and Prevention, they recognize this as well. And as part of the Healthy People 2030 goals put out by the Office of Disease Prevention and Health, their goal is for USDA and FDA to reduce overall salmonella illnesses by 25% by 2030. Well, if you go back to the previous chart, you can see that it's 2024, and if nothing else, we've had a tendency to tick upwards, if not stay the same, and we certainly haven't gone down, right? So we have 6 years left to be able to hit our 2030 goal that USDA and FDA are tasked with hitting. And so prevalence has worked, we've driven prevalence down, but again, we need to do more from an illness standpoint. And so that's where USDA and FSIS are really taking a different approach to how they're looking at salmonella in these fresh poultry products. And they're taking a look in a couple of different ways. And one is based on the overall final product risk to the consumer, and then also looking at it in terms of an overall industry impact. And so two very recent, what I would say are sort of landmark regulations USDA has put out just in the past year, where we're starting to move from salmonella being bad to now salmonella being an adulterant and bad. And so the first one is the salmonella as an adulterant in not ready-to-eat breaded stuffed chicken products. And then the second one is the salmonella framework for raw poultry products. And so the first one we're going to talk about is one where we're dealing with a specific product and a product type. And so this regulation came to be sort of as a result of a Salmonella enteritidis outbreak in 2021. There were 36 illnesses. A third of them ended up being hospitalized. And it really came from one particular, while this one came from Serenade Foods, which is a subsidiary of Maple Leaf Foods, it came from these different product types. And so they're very specific in the overall product type. And what you're seeing is that these are these stuffed items. So this is a product that's breaded. That's kind of got a raw chicken on the outside, and then some sort of a filling, like a cordon bleu or a broccoli and cheese in the center of the product. And so many of these products are sold raw. And there's been a number of outbreaks in these types of products going all the way back to 1998. To the clip of about every year and a half, there was an outbreak in these same types of products, right? So in 2015, USDA actually came in and changed the entire the labeling regulations for these types of products. So something that looks cooked but isn't. And they made different concessions on the front of the package where you had to specifically call out whether or not the product is raw. It had to be within the primary label. It had to be a specific font. You had to call out many of these different things on the package label. And so these are all the places on the product that was recalled where it was called out that, number one, the product is raw. Number two, you needed to cook it to a specific temperature. Number three, you weren't supposed to microwave it. And number four, you're supposed to use an oven, right? So these are so they were doing it right in terms of the labeling, in terms of informing the consumer, the proper ways to cook it, the proper endpoint temperature. They were doing it right. But in the end, this is what CDC and FSIS found, was that, number one, people didn't follow the cooking instructions. Obviously, right? That's why we're in the problem that we were. And they did it in a number of different ways. They use a microwave or an air fryer. So this is important. Microwaving a frozen piece is very challenging. Same with an air fryer, which works basically as a convection oven. The other thing that they did is they cooked for a shorter time than recommended when they did use a conventional oven. How many people here actually preheat their ovens? Oh, come on, be honest. Really? I'm impressed. Nice, that's very good. I don't. Like I said, I'm a thermal process engineer, and I don't preheat my oven. But that's important because a lot of the cooking instructions that are actually put together that are validated for these types of ovens, that is part of the actual cooking instructions, okay? And that is factored into the amount of time that they put on for the overall time that the product is supposed to cook. So many people didn't cook it. If it looked hot, pulled it out of the oven. Lastly, people didn't use a thermometer, right? And of the 60% of the people who actually own a thermometer, less than 10% of the population actually use it, right? So even if people had it, very few people use it, and I am just as guilty of that as anybody. And so that's what led to basically a final rule being issued in May of 2024, listing salmonella as an adulterant for the first time in not ready-to-eat breaded stuffed chicken products. And as I said, there were 14 outbreaks and 5 recalls going back to 1998 that led up to this. So it wasn't just the one recall in 2021. This had been a habitual cause. And the way that it reads within the reg is that lots that test positive for 1 CFU per gram or greater of salmonella must be diverted for other uses or destroyed, right? So there's not a lot of ways that you can divert this type of product. It's either good and you can use it, or it's bad and you have to destroy it. There's not very many ways you can divert at that point. And so again, this last point is key is because they're calling out salmonella, which includes any serotype, is an adulterant in this type of product. Okay, so again, we're looking at specific risk-based look in a particular product, getting us to salmonella becoming an adulterant. The second sort of groundbreaking, I would say, regulation that has come is the Salmonella framework for raw poultry products. And so this is something that is industry-wide within poultry. It involves turkey, it involves chicken, and this was released just recently in August. And right now the comment period is up until November 7th. So right now we're in the comment period and making responses to it as we speak. But the way that this looks is that there's actually 3 components to this framework. The first one being component 1, which goes back to the farm. So this is requiring incoming flocks to be tested for salmonella before entering an establishment. Second component deals more with process control monitoring at the plant to be able to make sure that your plant is in line with what it should be doing, okay? And then component 3 is implementing an enforceable final product standard. So this is what gets to salmonella being an adulterant in raw poultry products, okay? Component 1, really from where it began to what actually made it into the rule, has changed quite a bit. There was a number of things that were added that have been pulled out. And I think some of it goes back to just how much power does FSIS have to go back to the farm. They can make recommendations, but they can't regulate on the farm. And so that became, I think, a bit of a, if we want to really move this regulation through, that was a battle that maybe FSIS didn't want to fight at the time, is maybe the way to think about it. Component 2 is kind of a question mark. There's a lot of different things within component 2 that really make this a bit of a challenge from a processing standpoint. If you were going to use salmonella as your measure and you don't really have high amounts of salmonella, is that the best statistical process control for your plant? And if it isn't, and you need to measure something, now you're using something like Enterobacteriaceae or using aerobic plate count, which if that is what you're using, you're not necessarily tied to those measures being actual predictors of salmonella within your plant. And so this one is still up for debate. It'll be curious to see what the questions that we put together and what FSIS's response to that will be as far as where component 2 lands. Component 3 is something we're going to have to deal with, is probably the best way to put it. And so the way that component 3 works is that it's written as such, is that poultry products containing salmonella at greater than 10 CFU per gram. So now we're getting into load, the amount of salmonella within the product, and a serotype of public health significance. So you need to have load and a specific serotype is injurious and unfit for human food. And this is important. The language here is really important because this is the exact language that was used back in the late 90s when it was determined that E. coli 0157H7 was considered an adulterant. There's a bunch of different types of E. coli's out there. There's only a certain number of them that cause illness. This is the exact same language that went through the legal scrutiny that ultimately allowed 0157H7 to be considered an adulterant. So that's where, from a legal standpoint, it's going to be difficult to challenge this, I think. And that's why this one is likely going to go through. And so this gets tricky because as you kind of start to talk about, okay, what does this look like within poultry versus what is it going to ultimately look like in pork? You see between the three targeted serotypes, Enteritidis, Typhimurium, I4512I negative, monophasic, that's different than what is being asked for in turkey, right? So Hadar, Typhimurium mentioned, okay? So it becomes, now you're down to specific serotypes within specific proteins. These are the ones that are being targeted. And these only reflect the major causes of illnesses within these proteins over the last 3 to 5 years, okay? So as you start talking about what is it going to look like in beef, what is it going to look like in pork, these are the sort of things that are going to have to be fleshed out through risk assessments to be able to determine, okay, what are the three of target? As we've learned today, I4512I negative, probably going to be on the top of the list. But the other two might be interesting to see where that lands as it relates to pork. And so again, we're in comment period right now until November 7th. That may change. There is some push to be able to extend that out to April of 2025, which would be great. However, that is up to FSIS as to whether or not they will accept that. But as of right now, we have until November. And if that holds, final ruling in poultry is likely to be implemented in 2025. And we have been told by the Deputy Undersecretary that pork is next. So this is going to be something that we're going to have to be dealing with probably in the not too distant future. And so what do we know about pork in terms of Salmonella actually in meat? And I think that, this is probably a slide that I think a number of you have probably seen, but this is the actual, Salmonella isn't just a meat industry problem. I mean, it really truly is a food industry problem. And then if you look at this, 75%, almost 76% of Salmonella illnesses come from 7 different food commodities. And when you look at it across these, when you look at it across the 76% of the commodities, it's almost even in terms of the number of illnesses that come from both meat versus what comes from fruits and vegetables. So again, this is a food industry problem, not just a meat industry problem. But if you look, pork is a number 3 food commodity associated with Salmonella illnesses. Chicken is first, especially for those in the back that may not be able to see this. Chicken is first at about 17% of the illnesses attributed to chicken. Pork is right in there with fruits, seeded vegetables, and turkey at about 12.8%. And so again, probably one of the biggest industry-wide studies looking at prevalence across the meat industry was a USDA study that actually was done, I believe, in 2018, published in 2020, where they looked at the overall prevalence of Salmonella in large manufacturing facilities, small and medium-sized manufacturing facilities, and they sampled a number of different types of products. So they looked at comminuted, which would be like ground pork. It could also include bratwurst, could also include fresh breakfast sausage, anything that's basically ground, and they sampled the finished product. The other thing they looked at was intact and non-intact, so this would be like whole, this would be like ribs, this would be tenderloins, this would be boneless loins, this would be marinated tenderloins, marinated meats, different things like that. And so as you can definitely see, comminuted definitely sticks out in terms of the overall prevalence of Salmonella in these different meat types. 21.2% positive in comminuted. That number drops when you start looking at intact and non-intact. So whole muscle does a bit better in terms of Salmonella. Comminuted, definitely higher overall percent positive. But that was basically going in, taking a sample in a facility, and I can tell you right now that there are certain raw materials that were over-tested within that study for reasons that are fairly simple. If you're going to go into a plant and you're going to take a sample, put it in a box and FedEx it to a lab, how many whole Boston butts are you going to pull, right? When right next to it on the loin boning line, you might be able to take a tenderloin that's much smaller and easier to fit into a FedEx box and ship it. So there are certain raw materials that were oversampled within that period. But again, they took samples from a plant on one day, sent it, and that's what the overall prevalence rate is. So that leaves a lot to be able to understand, especially as a processor, to try to understand just how Salmonella essentially moves through my supply chain, right? To be able to understand, is prevalence and load consistent across processing dates and processing lines, okay? What serotypes are present, and are they consistent as they come to our plant? And that do these serotypes share relatedness? If we see it I4, 512, I negative, is it the same on Monday as it is on Wednesday? And so that's what our research aim was. It was to determine the Salmonella profile of two different raw materials and examine the relationship between Salmonella, the production, and the production environment within a production schedule. And so these are the two processing lines that we sampled. So if you look, so we looked at the picnic shoulder line as well as the belly line, basically. And so so we sampled the trim that came off of each one of these lines. So for the picnic shoulder trim, as you can see, there's sort of a line of people. They're basically using whizzer knives to be able to trim that hole or that Boston butt right there. And you can see sort of where they're trimming. So they take where they go through, they cut that trim off. And so all of that trim then basically drops onto a little conveyor, which then gets brought to the chute, and right there it sort of accumulates, and that's exactly where I took my meat sample as well as environmental swabs at that same location. On the belly line, basically what this is that we're using a water jet knife to make two cuts on the belly to right size it for further processing. And so what I'm doing, the cut that we are taking here for the what we call boot jack trim is basically the flank end of the belly as it's being cut. And so samples that we took here, we did it 3 days a week, Monday, Tuesday, Wednesday. We did it at the beginning of shift, so right away at the beginning of the day, about 6 o'clock in the morning. And then we also did it at the end of shift at about 1:30 p.m. in the afternoon. And we did it for 6 consecutive weeks through the months of October and November of last year. And so the way that we took the samples, I think, is important to understand. Really, we wanted to try to find as much Salmonella as we could. Was basically what we were setting out to do. And so we used a couple of different sampling devices that are a little bit different. The Easy Reach split sampler that is used is basically two sponge swabs together that after it allows you to swab the same area. And then once you're done, you can split that swab, go into 2 separate roll pack bags, and now you can do sort of a paired sample, so you can send that swab to Salmonella testing, or in our case, aerobic plate count and 16S sequencing. Now for the meat samples, we did something a little bit unique, and that's where we used the microtally mitt, which is being used throughout the industry. It's basically a cloth that basically you can use that goes over your hand. It's really good for if you want to take like carcass samples to be able to wipe down a carcass. It's much better than a swab to be able to do this. However, the way that we did it was a bit unique. And so when I would go into the plant, I would go in and I would take a 2,000 gram sample of the meat and then bring it back to the lab, and I would take all of that meat sample, put it into a sort of this oversized bag along with the mitt. I would then take it and shake it for 1 minute to be able to mix that meat along with the cloth to be able to get it all exposed to every surface in there. So that provided a number of benefits for us to be able to try to find Salmonella, because right now, if we were to do this, we were limited to a 375 gram sample. This allowed us to analyze 2,000 grams of sample and really try to cast as big a net as we could to try to find Salmonella. And so in the end, instead of, what we were actually going into enrichment with was the cloth at that point. And so from there, we would use the Bax Hygiena CellQuant method for the detection and enumeration of Salmonella. If it was positive, we would then go into a secondary enrichment to be able to isolate Salmonella from that. If it was typical black colonies on XLT4 plates, we would pick 3 colonies that we would then move on, freeze, and then that was part of what we used for whole genome sequencing and phylogenetic analysis. And so here's what we found. So we found, first off, Salmonella has a tendency to cluster by processing date. And this is something we've seen within poultry. This is also something that we saw in a pilot study that we had conducted the year earlier. And so if you look, so you see that you have 4 dates that we actually started, these 4 clusters where we started to see Salmonella. On the other days, we didn't see any Salmonella, okay? So next, so the other thing that was interesting with this is that we did not see it in the environment very much. It almost exclusively came in on the meat. We only had one particular day at one particular time on October 23rd at the very beginning of the shift where all 3 environmental samples were positive. For the whole rest of the study, 200 swabs, none of them were positive, okay? So the overall prevalence that we had was about 11.5% in both the bootjack trim and the picnic trim. We had no positives in the bootjack trim environment, and we only had those 3 samples in the picnic trim contact surface environment where we had positive Salmonella. When you start looking across serotypes, what we found, so we saw 5 individual serotypes present throughout the duration of the study. And as you look, you start to see that these serotypes also have a tendency to cluster by processing date as well. So you look, you see that in that first week, you see Salmonella London present on the 1 day. And then you look, the following week, we had a whole day where we found I4512I negative all throughout the production shift. And this was across multiple fabrication lines, beginning and end of shift. And it actually carried over into the 24th as well. And then on the next week, on that Tuesday, we had a number of Salmonella positives that involved Salmonella London again, Typhimurium, and Agona. And then the following week, we had another cluster that involved Agona, Enteritidis, and London. And so this is where we wanted to really try to understand, okay, so within these serotypes, how related are they? Are we seeing the same strain, okay? And so this is what we saw. So again, so this is a phylogenetic tree looking at all the different isolates that we had that identified as I4512I negative. So I want to also, just kind of go back to that previous slide. We only saw I4512I negative on 1 day, or actually on 2 days, but one particular cluster. And so here's what we saw. So this first clade that we had definitely had a completely different antimicrobial resistance, plasmid, virulence gene profile than the other I4512I negative strains. And as you can see, the other thing, it was carrying quat resistance genes, which as a processor, that's important to me because quat is one of the most used sanitizers that we use within the plant, okay? So the other clade that we had was also highly related. So 12 SNPs away from this sort of major grouping. But as you can see, it's carrying a couple of different plasmids that the rest of the group is not. So that did, while it's highly related, 12 SNPs away, it still did have a different profile than the rest of the other strains. Now the rest of the strains that we saw, 0 to 2 SNPs away. So these strains are highly related down to the sequence level across multiple days, multiple fabrication lines, and I should also add that these fabrication lines are on 2 opposite ends of the plant and actually on 2 actual individual floors within the plant, so they're not adjacent to each other at all in both the meat and the environment and at the beginning and end of shift. So these strains persisted or were continually brought to the line is what we saw. Again, looking at the Salmonella London strains that we had, again, very highly related, 0 to 1 SNPs away. I should also add that from a reference genome standpoint, I'm actually using one of the strains in one of my samples as the reference genome. So these are from an alignment standpoint that we're aligning all these sequences to, these are as close as what you're going to get to be able to see any of these differences, and they're still coming up highly identical. And that's across multiple days, and in the case of Salmonella London, we had Salmonella London on October 9th, October 31st, and November 6th. So over the course of that 5-week period, all of the Salmonella London we had was highly related. And again, it was seen across both fabrication lines. Only found in the meat, as I said, we only had the few samples that were positive in the environment, but also at the beginning and end of shift. Now this same relationship, this highly relatedness, although I'm not showing it, highly related for Typhimurium and Agona and Enteritidis, the other 3 serotypes that we had within this. In each case, they were all highly related, less than 3 SNP differences away. The other thing that we looked at again was that it was again overall prevalence. I mean, that was what we used to determine what we went back into to be able to try to pull our cultures back out. There's a couple of things with this. First off, overall CFU per sample, the overall load of Salmonella was low. Going back to that less than 10, if that's where we end up in pork, 93% of our samples were below that. 7% were at or above that. But the other thing that was really interesting with this is that since we were going back into that primary enrichment, because we wanted to get the isolates out, we had approximately 50% of our samples that were positive off of the Bax Hygiena system did not confirm culturally. So you can take that one of two ways. You can either look at those as false positives, or that we were or that there's a lot of information that we are missing with that the Bax Hygiena system picks up, that because of culture confirmation, we're not able to get to. So from a whole genome sequencing standpoint, we might be leaving a lot of data on the table for us to be able to bring that in to our analysis. So there's a couple of ways to look at it. I'm trying to be really diplomatic with how I talk about that. But anyway, so what did we learn? What did we learn? Salmonella prevalence has a tendency to cluster by processing date, in that we saw higher prevalence of Salmonella in the meat as opposed to the processing environment. To me, that's a big hint as to what potential sources for Salmonella in the meat may be. Salmonella serotypes have a tendency to vary, however, but they also tend to cluster around these certain production dates. Lastly, the individual strains isolated within each serotype are highly related across a production schedule. Again, taken in total, I think this would suggest that there's a common source, especially with the level of relatedness involved with these Salmonella strains. And then lastly, the potential strain-to-strain diversity within the samples that are highly related at the SNP level. As I showed, even though some of these strains were 0 to 2 SNPs away, there was genetic variation in sort of that ancillary genome of some carried 1 or 2 genes with it, even though they were highly related. And so the other thing that I want to bring up is that there are things that we do as a processor that we don't need whole genome sequencing to answer. You know, the top 2 things, I don't need whole genome sequencing to be able to say whether what serotypes I have and do they land on particular dates. The last 2 bullet points, I need whole genome sequencing to answer that. And I think that whole genome sequencing provides a great deal of the context for us to be able to understand back to where some of this Salmonella may be coming from. And so takeaways for the presentation, first off, USDA FSIS is now taking a risk-based approach to Salmonella. And as I said, this will be applied to pork probably within the next 2 to 4 years. The regulatory pathway to Salmonella as an adulterant is evident, and as I just said, it will not be limited to poultry products. I mean, so this is going to be coming in pork. And that honestly, further research is needed to understand sort of these temporal dynamics around processing schedule and the relatedness of Salmonella throughout the entire fresh pork supply chain. So with that, really appreciate everybody sticking with me at the very end. Hopefully I will be able to get everybody to the bar in time. But again, thank you.
Thanks, Aaron. Questions? Yes. Great.
Thanks. Why did they set 10 grams or 10 CFUs per gram? What is the...
Good question. So there were 2 risk assessments that were done as part of that reg. One was done to basically establish which serotypes were the ones of particular concern. The other one basically looked at if you were to, based off of the number of illnesses, if you were to if you were to take the high level samples out of the population, how many illnesses would that basically remove? There was a big discussion within because the way this was originally proposed, it was at 1 CFU per gram, which that's, from my standpoint, that's really scary, especially when I show data around the whole false positive or the potential, or gotta stop saying false positive, the ones that did not confirm culturally. And so that's, 1 CFU, so that moved to be able to say that, based off of that risk assessment, that 10 CFU really was effective at reducing what would be predicted as illnesses. From a processor standpoint, that was really important, but there was also definitely a public health aspect to that as well. Yep.
I have a question with the Salmonella prevalence, the study that you presented, the 3 types of meat, intact, and then is it that the intact and the non-intact, when they're in single digits versus the minced one, is more to do with the sample being processed, because the intact, you don't have much surface area release, and the Salmonella is intracellular. So is it like we are underdiagnosing the numbers of CFUs of Salmonella with intact and non-intact versus with the minced one?
I'm not exactly sure. It's an interesting idea. I think, I would say that based off of prevalence and some of the things that we've learned, especially around lymph nodes, that there's certainly a greater potential for lymph nodes to be brought into ground. Now that doesn't mean that that's the only source for it, but again, you have to remember that they were sampling raw, finished products. So, for example, I was testing trim components, right? The belly trim, the picnic trim. Ultimately, those end up in a larger blend with a bunch of other types of trim to be able to get a target fat percent. For us to be able to be at a 72% lean trim, which goes a lot of different ways. So now you've compounded that and spread it from a overall prevalence standpoint. So if you would have had a hot day on, that would have then, so you can kind of see where in the finished product you can start to get higher overall prevalence than maybe what you see in the individual trim components. So maybe that had a little bit to do with with it as well. But I think based off of what we see when we are doing our own testing on this, is that 21 to 30% number isn't real far off. It's probably a pretty real number, and it can vary by processor. So, yes, sir.
Here, great presentation. Curiosity on the non-culturable status. Salmonella, we know, can enter the metabolic status of being viable and non-culturable. Have you tried different media to see if it truly wasn't? And then, have you thought about doing a PCR to assess if it was live or dead too, those different types of PCRs?
Yeah, so good question. When we were in the midst of running this, we're kind of having to run all of this in real time. So we didn't have a lot of opportunity to really dive into, whether or not these were true false negatives or not, or false positives. I can tell you that on 6 of the samples, we did do further work on the primary enrichment, looking at those particular samples across a number of different secondary enrichments as well as selective media for plating. All of them came back negative. So while I can't necessarily say that every single one of them were necessarily non-culturable, I can say for at least those 6, the 6 out of the 6 that I did do further testing on, they were true false positives.
All right. Thank you so much for attending, and have a
Thank you.
great time.